Hey everyone, I'm Dr. Anya.
And I'm Dr. Ben.
And today, we're doing something a little different. We're going to ditch the textbooks and take you on a journey, an incredible journey. We are shrinking down to explore one of the most amazing places in the universe, the living cell. You ready for this?
Okay, Ben, you got the miniaturizer ready? All systems are a go, Anya. Engaging the shrink sequence. Now. Brace yourselves, because from our perspective, everything is about to get huge. Here we go. Whoa, we're in. We made it. Look around you. It's incredible. Okay, so right in front of us, that massive, bustling structure, that's our destination, a eukaryotic cell. It looks just like a tiny city. But hang on. See that smaller, simpler-looking thing floating nearby? That's its older cousin, a prokaryotic cell. The difference is just, wow. Let's break it down.
All right, so let's get our bearings here. On one side, you have the eukaryotic cell, our complex city. See all those different buildings and departments? Those are membrane-bound organelles. And it has a true command center, a nucleus. Now, look at the prokaryote. It's more like a one-room workshop compared to a sprawling factory. There's no separate office, no nucleus. Its genetic material, the DNA, is just kind of bundled together in one area we call the nucleoid.
Okay, and check this out. See these little folds in the prokaryote's membrane? That's something called a mesosome. Now, for all you NEET aspirants out there, this is a super high yield point. Think of the mesosome as the prokaryotic version of mitochondria. It's not the same structure, but it has a similar job. It's all about increasing the surface area for really important reactions like respiration. It's a key adaptation.
Okay, tour's moving on. Let's head toward the border of our eukaryotic city. We're approaching the plasma membrane. Now, this isn't just some static brick wall. Oh no, this is a dynamic, intelligent gatekeeper, a fluid border that's always active, controlling every single thing that comes in or goes out.
Just look at this surface up close. It's absolutely beautiful. This is the famous fluid mosaic model, live and in person. You see how those large structures, the proteins, are just floating around? They're like icebergs in this shimmering sea of lipids. The whole thing is constantly in motion. It's not rigid at all, it's fluid, it's alive.
Anya's spot on. And the secret to this whole fluid structure is the design of these little molecules called phospholipids. See, they have heads that love water, so they face outwards to the watery environment outside and inside the cell. But their tails hate water, so they tuck themselves away on the inside, creating this oily layer. It's a simple but brilliant design that forms a perfect, flexible barrier.
Okay, we've passed through the gate. We're inside the cytoplasm. Whoa, it is busy in here. Just look all around us, this massive interconnected network of membranes. This, my friends, is the industrial heartland of the cell. We're talking about the endomembrane system, the factory, the packaging plant, and the shipping department all rolled into one.
So let's trace the journey of a brand new protein. It all starts here, at the rough ER, where it's first synthesized. Think of it as the main assembly line. From there, it gets sent over to the Golgi apparatus. That's like the finishing and packaging department, where it gets modified and sorted. Finally, it's packed up into a little transport bubble, a vesicle, and shipped out to wherever it's needed. The logistics are just mind-blowing.
And as we float past the ER, you can actually see two distinct regions. Over here, look at that bumpy, studded surface. That's the rough ER. Those little studs are ribosomes. They're the actual protein-making machines. Now drift over here. See how this part is all smooth? That's the smooth ER. No ribosomes because its job is different. This is where the cell produces lipids and handles detoxification. Two parts of the same factory, but with totally different jobs.
You know, I can feel a sort of hum in the cytoplasm, a real buzz of energy. A city this busy must need a ton of power. Let's head over to the source. Time to visit the power plants.
And there they are. Look at that sausage-shaped organelle. That's a mitochondrion. This is the absolute powerhouse of the cell. It's constantly burning fuel through cellular respiration to produce ATP, the cell's energy currency. And if this were a plant cell, we'd also see these incredible green discs, chloroplasts. They're basically microscopic solar panels capturing sunlight to make food. Two amazing energy converters.
But Ben, here's where things get really, really weird and cool. The going theory, the endosymbiotic theory, is that these organelles weren't always part of the cell. Billions of years ago, they were their own free-living bacteria. Think about it. They have a double membrane, like they were swallowed whole. They have their own circular DNA, just like a bacterium. And they have their own ribosomes, the 70S kind, again, just like a bacterium. It's literally a cell living inside another cell. Mind-blowing, right? All right, time for the main event. We've seen the factories and the power plants, but now we're heading for the C-suite, the City Hall, the absolute command center of this whole operation. Let's go visit the nucleus. And here it is. Wow, it's enormous compared to everything else. This entire structure is protected by a double-layered wall, the nuclear envelope. And see all those little gateways dotting the surface? Those are nuclear pores. You can think of them as the high-security checkpoints. They are super selective about what gets in and what gets out. Nothing passes without the right credentials.
Let's peek inside. That dark, dense region right in the middle, that's the nucleolus. Its whole job is to be a ribosome factory, churning out the parts for those little protein-making machines. And all this stringy, tangled stuff filling up the rest of the space, that is the chromatin. It's the cell's DNA, all wound up with proteins. This is it, the master blueprint, the entire library of instructions for everything this cell will ever do. Okay, our tour inside the cell is coming to an end. Let's start pulling back. But wait, before we go, there's one more secret to uncover. As we zoom out, look at the cell's outer surface. We've seen how it works, but how does it move? The answer is just a masterpiece of engineering. Okay, get ready to zoom in. We're looking at a cross-section of a cilium, one of those tiny hair-like whips on the outside of the cell. Now, it might just look like a simple hair, but when you slice it open, look at that. The inside is this incredibly intricate, almost gear-like arrangement of filaments called microtubules. It's stunningly organized. And here it is. This is the money shot. This is the pattern you have to remember. Count them with me. You have 9 pairs or doublets of microtubules arranged in a perfect outer ring, and then you have 2 single microtubules right in the center, 9 plus 2. That is the classic textbook 9 plus 2 arrangement that forms the core of cilia and flagella. This is what powers their movement. Burn that image into your brain. And we're back to full size. What an incredible ride. Okay, before we sign off, let's do a quick fire recap of the absolute must-know high-yield facts for your exams. Ready? Fact 1. Ribosomes are non-membrane-bound organelles. Super important distinction. Fact 2. The mesosome in bacteria is functionally analogous to mitochondria. Fact 3. Know your ribosome sizes. Prokaryotes have 70S ribosomes, which are made of 50S and 30S subunits. Eukaryotes have larger 80S ribosomes made of 60S and 40S subunits. Got it? Good. So we've just toured the intricate, complex city that is a single cell. And it really makes you think, doesn't it? If one tiny cell is a whole metropolis, what does that make a multicellular organism like you or me? A nation? A world made of trillions of these cities, all communicating and cooperating? It's something to think about. Thanks for shrinking down and joining us on this amazing journey.